Now in this video we are going to talk about limbus, which is actually the corneoscleral junction. For example, if we look at here, we can find this cornea and this is our sclera, and the junction between the cornea and sclera, this over here, it is known as the corneal limbus. Now the histology of the limbus, it is very much important. First of all, it is about 1.5 millimeters wide band of the tissue that lies between the cornea and the sclera. Now the limbus is a very important structure because first of all it is going to have these connections. First of all, the corneal epithelium, it will be converted into the conjunctival epithelium. The first or the anterior limiting membrane of the cornea, it becomes absent. That Bowman's membrane, it is not present at the level of the limbus. Secondly, the sclera becomes continuous. The sclera of the cornea becomes continuous with the sclera proper. The stroma of the cornea becomes continuous with the sclera proper. Then the third thing is that endothelium, this one, and the posterior limiting membrane or the descending membrane, they both form this trabecular meshwork. So if you remember this diagram, you can remember that the corneal epithelium becomes conjunctival epithelium, the Bowman's membrane disappears, the stroma becomes continuous with the sclera proper, and the endothelium and the posterior limiting or descending membrane, they become trabecular meshwork. So what is a trabecular meshwork? It is actually composed of these canals, which are actually endothelium lined, and between these canals is present this connective tissue. Now what happens, there is a lot of aqueous humor. So this aqueous humor moves through these connective tissue lined vessels or this trabecular meshwork and moves into this region, which is known as the canal of Schlemm, which is actually a circular ring-like structure. And on the posterior side of the canal of Schlemm, where there are a lot of connector vessels, and these connector vessels connect this canal of Schlemm to the episcleral veins. In this way, the aqueous humor moves through this trabecular meshwork into the canal of Schlemm, and through these connector vessels, it moves through these episcleral vessels or veins. This is how the whole method is applied over here. The whole thing written over here in this paragraph has been explained through that diagram. Now the canal of Schlemm, how it is connected to the episcleral veins by the connector vessels, each and everything is written, and how the aqueous humor drains through the trabecular meshwork, through the canal of the Schlemm, to the connector vessels, to the episcleral vein, all each and everything has been mentioned. So this is our limbus, and main thing about limbus is that it is a reservoir of the limbal stem cells. Now these limbal stem cells, they will go to that corneal epithelium and they will form the basal layer of the corneal epithelium, which will actually have proliferation and will result in the regeneration of the corneal epithelium in case of any injury or damage or death of the corneal cells. In the next video, we are going to discuss our sclera.